Test, test. Is my voice clear?
Yes, sir, we can hear you.
Okay, please grant me the
Yes.
present. Okay, thank you, Fadin. So, good afternoon, everyone. So, let's continue our progress in learning together. So, this is the first time we implement active learning. So, everybody is learning because we are writing books. And I hope all of you will survive in this course. And because the target is like this. So we will complete chapter 12, including functional genomic, during our midterm semester. We are in chapter 5 now, okay? And after midterm exam, we will continue how to modify Nextflow. So we have to understand Nextflow pipeline and change some code inside from Python to Rust to improve the performance, concurrency, and parallelism. This is in part 4, more practical, and how to embed machine learning in the pipeline, because some of the pipeline require predicting a value like regression or doing classification. So we will cover also this one. And part 6, mostly on looking on the tools and trends in the future. I hope we can complete this book by this semester, and you will be very proud because you are part of the process. And we will keep improve the book as an open source for the next students who take this course. And also we will keep improving the quality of the writing and the code itself. Okay. So I got message from Professor Al-Hadi yesterday that you need to have assignment, but for me, your assignment is here. I will make sure that every one of you actively contributing to this chapter. So based on Majere feedback last week, we switched chapter 1 to chapter 2 and chapter 2 to chapter 1. And Fadin have some changes here. You are free to give feedback or change the text, especially if there is a redundant text or repetitive text. Just change, yeah? So just edit and change. And if you're more interested on the code, then make sure that you do the code QA and running the code with QA, like you run on your machine and then put some dependency like this. Okay. So please help me to make sure each chapter, the code is tested, the text is proofread, and we are now in chapter 5. Okay. I'm here in chapter 5 and 6. I think I'm done the code generation from ChatGPT o1 Pro, but we need to proofread, redundant explanation. We need to do, if you find some concept that is hard to understand by only the text, then find out good picture in Google and embed into the text and give some explanation. Let me give you example. So let's say we are talking about sequence analysis, right? In here. So you can go to Google Image sequence analysis in bioinformatics. If you got a good image that illustrates, maybe like this, okay, that you can align to the text. So you can put, let's say I like this picture, and then I download NGS map, and you can use GPT. Let's say you only have access to GPT-4, and then you can upload the image, and you can say, give a sentence caption for the image and explain the image in one paragraph, okay? So you can give this to the caption, and then you can take this, modify little bit, align little bit, and put on the text, okay? It's very easy to put image on the text. Just slice. Let's say I want to put the image here, okay? Slice image and upload, then just image, and then give the caption. Okay, and then you can pass the explanation. Okay, but as this is just example, I let it to you. I will delete here. So make sure that you read, proofread, and if you find good image, feel free to add image. That is the comment that I put here, okay? And if you're more interested on the code, then help me to do the code, compile. If you are done, then give additional dependency on the code for the dependency here. That means the code is tested. Okay. So let me briefly introduce the content of chapter 5. So after we cover introduction to RAS and RAS with bioinformatics and genomic and data structure for bioinformatics and genomics, now we are entering chapter 5, which is some of the analysis in bioinformatics. Not necessarily functional genomic, because we will cover in the chapter 9 to 12, but mostly about the bioinformatics itself, okay? So it starts from the reading of the sequence. Each machine has different precision, different error rates, different price as well. So the best is actually Illumina, PacBio, and the last one is Oxford Nano 4. But as you can see here, the raw error rate reaching 5 to 15%. This one is only 0.1 to 1%. Okay, quality come with the price. Of course, Illumina is pricey compared to Nano 4. But sometimes we don't need that precision. Sometimes we can use Nano 4 or Bioscience, okay? Once you have the machine, of course, it works like a physical instrument that reads the sequence, okay, with different methods, of course. But as a bioinformatician, you need to understand how to get the data and where to store in the computer. So previously we learned about Bloom filter. So Bloom filter is actually probabilistic data structure to capture stream of data with intention that you want to differentiate unique sequence, you can search unique sequence inside the data structure. But because this data structure is already covered in previous chapter, we focus on another one called hyperlock. Okay, hyperlock is another efficient data structure to read the data from the sequencing devices. Okay, so in this chapter, it's more focused on hyperlock implementation, which is also a probabilistic algorithm, but with different kind of mechanism because this is a probabilistic data structure, means that it's not really precise, but has some estimation inside regarding the presence of the data sequence. So of course, it's quite different with the Bloom filter. So again, you can find, let's say, some picture if you want. Usually you can get it from papers like this. To give more explanation, feel free to add more picture to visualize. Otherwise, you have to visualize from mathematical equation. But what is hyperlock? Is imagine a value x, which is a long sequence. And then I hash it. Hash data structure is a mechanism to compress long string into smaller string, but you still maintain a mapping between the largest string into the smallest string. So if I say hash x, that means we hash a longer string into shorter string, and then and then it will compute the binary representation from the hash, and that is what they call it, hyperlock. Okay, there's many other picture here, but make sure that if you put a picture, it's well aligned with the text. Okay. So hyperlock implementation, of course, as long as you know the data structure, HLL implementation is very straightforward in any Gen AI like GPT. So using on Pro in GPT, this is the code that it generates. It creates the structure hyperlock. Okay. And then this is the implementation of the structure. And this is the small main code to illustrate how it works. Okay. We need to test the code, make sure it's running. And next is another example. Let's say we want to demonstrate high throughput sequence using concurrency, means that we want to parallelize or make sure that the code is efficient. So let's see, we have a FASTQ file that already compute basic read statistic. So in RAS, we have need detail is a crate for efficient streaming of large FASTQ data. So it's a very specific crate called need detail. And in this code, it show you how to read from command line, okay, of the FASTA file, and then you utilize the need detail instead of we implement by ourselves. Okay, there's another program also for multiple JSON file combined, and then we parallelize using Rayon. Rayon is a parallel computing crate from RAS. Okay, this code need to be tested again for the reading. Previous before us, there's so many C++ library. Let me give you example here called Booty. Bowtie, a Bowtie or Bowtie, I don't know how to mention, but it's someone else's name. Okay, so is a C++ code is a very fast and sensitive read aligner. So you can see a lot of code here in C++. Okay. Later, if you have time during your research, now it's very easy to port this to Rust. If you use AI, because Rust and C++ is quite similar language in terms of low level and system language. Why is not open? Okay. So AI like Cloud now can get input from GitHub and perform the porting. Okay. So analyze the following C or C++ code in GitHub for genomic realignment and provide recommendation on porting it to Rust language using best rate available. Okay, you can pass here and run the porting process. If you have paid Clouded, the process of porting will take some time, of course, but this is popular genomic realignment in C and C++. The key component is index, Sambam output generation, multi-threading, and then this is the recommendation of the porting. Core Bioinformatics use Rust Bio, need detail, Rust high throughput sequencing library. Sure. And then it will take some time for it to port. But as you can see on the right side, the AI is working to port the code from C, C++ to Rust. If you have professional version of this Anthropic Clouded, you can create another GitHub contain Rust version of Boti. Okay, I will run it until my token, free token, gone. But as you can see, it also gives you the package. So what you need to do is to change, to test, yeah, and to gradually test the code and fix the bug error, also using AI. So it's very easy now to port any library from one language to Rust. Okay, let me continue. So in the next section, how to perform quality control on the read sequence. So some reads are, of course, not all reads are high quality, but maybe you can find low quality of the reads. But how we can model the quality control of reading from statistical point of view, of course, we utilize some statistical modeling to how many reads may contain error or contamination. We utilize binomial models, let's say like this. And then this is very common mathematical approach. If you have data, normal data and abnormal data, or normal data and wrong data or low quality data. So you can estimate, you can utilize some probability distribution to say that this may be for the normal data, and outside this distribution is actually abnormal data. Okay, that kind of approach, let's say you can use binomial distribution or Poisson distribution, and then you can test the code in this chapter. It gives you the sample code implementation in Rust. Okay. Of course, this is not machine learning based, but you can always improve machine learning, adding machine learning into your RAS code using threads like Lympha, but we will cover machine learning in the part 5 of our books. Okay. Previously, before, there's also many code like Qt. How to mention this again? Yeah. So Q.apt. Yeah. So another code to remove adapter sequence from sequence trees. Yeah. Another sample code, but this one is built on language Python, yeah, and Cython. Okay. Don't worry if you find library not in Rust, as I saw you before. Porting to Rust is trivial in this AI era. Okay. Maybe you can write paper how to port, let's say, Booty to Rust and compare the performance. I think that is worth for IEEE Key 1 paper. And report the performance gain if you put on Rust. Yeah. Okay. So it's also discussed some searching algorithm like KMP. We discussed this before in the previous chapter. Knut-Morris-Pratt algorithm is a graph-based search for sequence. Okay. Next, we cover also in this chapter the alignment and mapping algorithm. Alignment allowing the scientists to pinpoint where the sequence read originated in a genome and enabling downstream analysis like variant calling or gene expression that we will cover in other chapter. Okay, but alignment algorithm covered here, let's say like Needleman Wunsch global alignment, Smith-Waterman local alignment, yeah, and Burrow-Wheeler transform techniques and FM index, and give the sample code as well for the alignment. The following section covered the de novo assembly approach. Okay, it's a very central role to reconstruct entire genome from what you get as a fragments of sequence tree. So if you have a fragment of sequence trees, how to combine it again, reconstruct again the long sequence. We call it de novo assembly. Assembly refers to the reconstruction process to assembly again the genome. Okay? Of course, there is data structure being used. There is algorithm being used in this chapter using the Brueggen and graph assembly concept in the De Brueggen graph. Okay, De Brueggen already covered in the previous chapter, so we can refer to that chapter, and this is the implementation. And then variant calling. So variant calling and genotyping is very important part of this chapter because this task is to identify the genetic variants that may be linked to some specific traits like disease or drug response or anything that is related to metabolism. So how we call the variant, of course, it's based on how we understand the SNP, yeah, single nucleotide polymorphism. As we discussed previously in the dogma, in maybe chapter 2, so we have the SNP, single nucleotide polymorphism, and also small insertion or deletion to detect with the accuracy. So variant calling is an approach to compare the observed sequence data against something known, yeah, in the database like a DBSNP or any database that is proven by other researchers. So how you compare your variant, your SNP to the proven one that people already know that this SNP is related to, let's say, some disease or some problem in drugs or some recommendation in drugs. Okay. So this chapter covered that variant called an indel, how to insert and delete from the variant call, and give you sample code based on the explanation. Okay. Next section cover about structural variant detection. Okay. So if the variant change or the genetic change, it can rearrange the whole genome. Okay. And something need to be insert, something need to be delayed, some duplication need to be removed, inversion, and etc. So usually we call it SV, structural variants, is quite different from small variant. It's not small variant like SNP and indels, but is a structural variant. Okay. So this is by convention, yeah, this is another variant, structural variant that we can analyze from our sequence. Okay. This is the sample code you need to test. And the last chapter, the last section in this chapter is about, not the last, RNA sequence and transcriptomic analysis. So from the DNA and then RNA and then protein, the process of transcription to protein, we call it transcriptomics. Okay. So RNA sequence and transcriptomics is like a collection of process from gene expression, splicing variants to regulatory mechanics. So there is some biological background here discussed, and then transcript assembly, and give you sample code as well as an illustration. And this one is the last one. It is integrating short and long reads. How we integrate some reads, both shorter and longer reads. Put in JSON and then merge everything. Okay, so quite long chapter, but it's very common task if you have machine, NGS machine, and basic task of reading the sequence and basic process of the sequence like variant calling and other process. So I hope you can continue based on this comment from me here. We need people to proofread, need people to add image. We need people to code review. After you're done, and then put done, and we will move next week to chapter 6. Okay. I believe we will have midterm semester in exam in April. So maybe we have 2 more weeks to reach, let's say, end of part 2. Yeah. I hope we can complete this part 2 before, and then for part 3, we will make it your midterm semester to complete the chapter. Okay. But I try to write it using GPT before the midterm semester up to chapter 12. So your task on the midterm is to complete the half book up to functional genomics. Okay, that's it from me today. So please take time to look on chapter 5. Chapter 6 is also done, by the way, but you can start, but we will discuss next week. This is about high throughput sequencing data analysis. There is a specific library called STSLib. So we will cover next week about chapter 6. So focus this next week about chapter 5. In the next Saturday, we will cover chapter 6. And hopefully we cover up to chapter 8 before midterm semester. And during midterm semester, I will give you tasks to test to complete the part 3 of the book. Okay? You can pick one chapter and working on it. Okay, that's it for today from me. Any feedback, any comments? I see some comments in the book. Okay, let me check. Trianto run all the code completing. Okay, good. So please help to make sure that the cargo TOML, we don't need the whole TOML actually, but we need the dependency. Which version of the threads are you using? Yeah. So like package name version, we don't need the whole, but the code need the dependency like this. Yeah. So you don't need to put the TOML, but make sure that this dependency when you compile is put here. Sometimes if I upgrade Rayon or downgrade Rayon, it's not compatible with, let's say, NDRI or RAND. Yeah. So make sure this compatibility of this dependency you put here when you compile the code. This is also, we need the, it has no threads, so no dependency. Okay, this is using 0.90. Okay, that's good. But also we need help. If you find good picture other than this, please put. Yeah. So some people like to do the code. Some people like to do the text. Okay. Make sure we are teamwork. So in chapter 5, one doing the text, doing the proofreading, one find the image from Google and give visualization to the text, and Trianto can help on the code side. And if Trianto can make sure the GitHub of the book is clean and well tested, that will be very good when we release the book. We also release the GitHub code. Okay. So we will release the book as an open source web book like this. So you will get help from runtime team to turn CurveNote into web book. So if you have the GitHub, that will be very good. And this book is sell in Amazon. Not really big money, but if you involve in the book, you will get the revenue share from the book if it's published in Amazon and Google. Okay? Any feedback, any question for chapter 5? Is clear enough?
Clear, sir.
Okay, let me check one by one. I heard that Jonathan is resigning from the course, but I still have here Majere, Trianto, and who is Zach? Jonathan?
I think Zach is the new student that Prof. Alhadi mentioned. Is that right, Zach?
Yes, yeah, I'm the new student from Prof. but
Can you still follow the course, Zach?
I'm still trying.
Yeah, maybe you need to have guidance from Trianto or Fadin. Yeah, please try. Don't take too many course because usually master degree, you can only work like 8 to 10 credits per semester. Otherwise, it's too much. Yeah. Spend enough time reading, reproduce the code, reading, reproduce the code. And if you contribute to the book, you can take this task like coding or proofreading. It's the same contribution. Yeah. We will make this book as our guide. We will use this book as our book. We will improve this book every semester. Okay. Who else? Majere. Majere.
Pak, good afternoon, sir. I'm here.
Yeah. So please help with chapter 5, yeah?
It's okay, sir. Noted.
Yeah, you can do images from Google or proofreading and three. Okay. Or
Okay.
so if we complete this book
Okay, Abby, thanks for coming.
for good score.
It's okay, sir.
Okay, good. That's it from me. Anything from Fadin before we close, please?
Yes, thank you, Mr. Resman, for the explanation of the chapter. I just want to remind you guys to fill the attendance form in here for each lecture. And for Zach, you can check on the description of WhatsApp group, and there is a link to do the attendance form in here. And also, I already checked the GitHub from Trianto and Majere, and very active, Mr. Risman here from Majere and also Trianto is already doing the chapter 4. And for Christian, I don't know, in the last
need-
two class, she don't then. Yes.
We need to help Christian and Zach to be active because there is no book in RAS for bioinformatics and genomic in the world. You are the first one if we release this semester, and you have to be proud of, yeah? So please activate Christian and Zach later.
Yes. Yes, sir. Zach, you can fill in your email and also create GitHub repositories like Vejere and Trianto. And I will check each of your works and it will be counted as the um weekly task for your final grade. And after all of the comments, and don't forget to give comments also, I will do the final QC and check on whether is it working or not for a chapter. For example, in here, you can see from chapter 1 because Mr. Risman renewed the chapter 1 in here to make it more beginner friendly. So Zach, it is the right time to see the chapter 1 because it has been adjusted to beginner friendly. You can try running from the start to create a new Rust project and how to run it. And I will check later on. And please help me also, guys, to give a comment if you have some errors or finding some interesting. And yeah, like Mr. Risman said, Cloud is pretty amazing also to do the quality check of the code and give recommendation what to do. Like before, I previously worked like the Qt adapt library all tools like and also Bowtie. It is pretty common in Galaxy project or Nextflow, but in here, yeah, we can try to porting it in also in the into the Rust because there is basic bioinformatics the first task to do before running a complete sequence. We have to check whether it's there are some not good quality of sequence or not good quality of reads before processing any more into the deep dive third analysis like variant calling, etc. I think that is from me, and you can also check in into the Notion page to deep dive more into what is Rust, what is Nextflow, what is GitHub and VS Code if you don't have it yet. That's it. I think we can meet again, Mr. Swan, in the next week, yeah, before before yeah, Idul Fitri, and I think the class is still running. in the next week, 29 March. Thank you, Mr. Usman. Is there anything that you want to add?
Thank you, Fadin, for your notion and guidance. Looks like Christian and Zach need more help. So Majere and Trianto, please help each other. Yeah. You are leading the test code and that's good for you. Yeah. Because if you look on the market, there is no one release book like this. So this will be smart for future researcher who are doing with us. And it's very good for you to be active on this in GitHub. Okay. By the way, a company like 10 Time Genomics start using RAS, and many other company, of course, will take a look on RAS. It's very good for your portfolio during this course. Also very good if you take your master degree topic on integrating machine learning, deep learning, or reinforcement learning into pipeline analysis in NF4. Is a very open area if you can combine Python and RAS to make it works in Nextflow. It's very easy to get Q1 paper. So what you are doing now is also doing your future paper. Okay.
Okay, sir.
Okay, sir. Thank you.
Thank you. That's all for this week. See you next week on chapter 6.
Thank you, sir.
Yeah.
Thank you all. Thank you, Mr. Usman.